Okay, so welcome back to this next video in which we are discussing the use of short hairpin RNAs to prevent the expression of a certain protein. Okay, so what we've seen so far is that we have produced from our short hairpin RNA this mature short hairpin RNA, which basically is a double-stranded piece of RNA where each of the strands of the double-stranded piece of RNA has an overhang basically of two nucleotides, and this is at the 3' end of that strand. Okay, now the double-stranded piece of RNA will then have another protein, it will have a protein bound to it which binds to double-stranded RNA, and this will either be the TRBP protein, which stands for the Transactivation Response RNA Binding Protein, or it will be the PACT protein, which stands for Protein Activator of PKR. Now, bound on top of whichever protein you have there, you then have this type 3 ribonuclease enzyme called DICER. Okay, right, so this is where we've got to now. Now, I think it's about time that I told you what the sequence of organic bases that we're going to have on this double-stranded piece of RNA is. Okay, so remember, for all of the stuff that we've seen so far, it did not matter what the sequence of organic bases was. Basically, the process would happen the same just as long as this second portion here had the same had a complementary sequence of organic bases to this first section, but backwards, of course. Okay, so providing you chose this second section carefully, it did not matter what the sequence of organic bases were on this first section here. You could make it up, basically. Okay, so now let's talk about what we're going to make that. Well, we are going to make it so that it is complementary to a sequence of organic bases on the piece of mRNA for the protein A. So remember, protein A is what we're trying to knock out. We want to stop the cell from being able to make protein A. Now, protein A has two genes, okay, and both of these genes will be making mRNA, okay, and this mRNA will then be translated into the protein A. Now, the mRNA here, so let's say this is the mRNA for protein A, this will have a certain sequence of organic bases. So let's make a sequence up, and remember, uracil replaces thymine in mRNA. Okay, so let's have AUG, CA, C again. Okay, right. What we are going to do basically is we're going to make this sequence of nucleotides here complementary to this sequence here. Okay, now let's say this is the 5' end of this mRNA for protein A, and let's say this is the 3' end. So basically, what we want is this sequence here to bind to a portion of this. Okay, so let's have our sequence here, and let's say this is the 3' end here, and this is the 5' end, just like I've got down here. And basically what we will do is make it so that it has the complementary sequence to this section of the mRNA of protein A. And I should make this clear that the mRNA for protein A could be massive, you know. It could be very, very long. You will only make this section complementary to a short portion of it, basically. Okay, because this isn't too long. This section that you're going to use in your double-stranded RNA portion of the hairpin, it won't be too big, basically. Okay, so let's make it U, A, C, G, U, A, G then. Right, okay, so this is now what we would pick as our sequence to put in position 1 here, and then we could design the sequence in position 2 so that it was complementary to this sequence but in reverse. Okay, so you now just replace all of these with these here. Okay, right. Now, let's discuss what's going to happen next. Okay, so let's go back now to our piece of mature shRNA. Okay, so we've now discussed that this piece of mature shRNA, it will have a sequence within this double-stranded RNA now that is complementary to a section of the mRNA for protein A. Okay, but at the moment it's still got this protein which binds to double-stranded RNA bound to it, and it's still got the dicer enzyme bound to it. So what's going to happen next? Well, basically another protein is going to come and associate with it, and these two proteins here, the double-stranded RNA binding protein, which remember is either TRBP or PACT. Okay, so I'll just label these back up again. So this is either TRBP or it's PACT. Okay, and we also have the dicer enzyme, which is an example of an ribonuclease type 3 attached. These are going to help with the loading of this new protein on. Okay, so what's going to now happen is a big protein is going to come and bind here. Okay, so this new protein, let's highlight this in a special colour because it's brand new. Okay, so this new protein has the rather fantastic name Argo02. Okay, so this protein is going to be called Argo02. Okay, and for short, Argo02 is often abbreviated to AGO2. Okay, now what's going to happen is that once Argonaut2 has bound to our double-stranded piece of shRNA now here, what's going to happen is the other two proteins, the dicer enzyme and the protein which is either TRBP or PACT, those two proteins are going to break off basically, and you're going to be left with Argonaut2 bound to this double-stranded piece of RNA here. So here's Argonaut2, and here is our double-stranded piece of RNA with the 2 nucleotide long overhangs on the 3' ends of each of these strands. Okay, so this is Argonaut2 here. And I'll colour it in its colour, so it was going to be represented in green here, like so. Okay, and I might even write AGO2 in that box since it's so large. Okay, right. So the dicer and the TRBP or PACT have gone off, basically. Now this complex of the double-stranded piece of RNA along with the AGO2 protein, this is what is known as the RISC. Okay? And the RISC stands for the RNA-induced silencing complex. Okay, so the R stands for the RNA, the I stands for induced, and then the S stands for silencing, and finally the C stands for complex. Okay, now what's going to happen is Argonaut2, basically it's a ribonuclease enzyme. It cuts up RNA, and what it's going to do is it's firstly going to get rid of one of these strands, basically. Okay, so it's going to break down one of these strands now. Okay, so it's got the double-stranded piece of RNA at the moment, and now it's going to get rid of one of these strands. Now, basically, one of these strands in this double-stranded piece of RNA was complementary to the sequence on the mRNA of protein A. So let's put the protein A mRNA up here. Okay, and basically one of these two strands was complementary. Okay, so it could bind to the mRNA of protein A. The other could not. Okay, so let's say that it is this one here, so let's colour it in turquoise. So let's say it's this one here. Okay, and this one cannot bind to the mRNA of protein A because it's complementary to this turquoise strand, and therefore it has the same sequence of organic bases as the mRNA of protein A, and therefore it can't bind to the mRNA of protein A. Now, the one which can't bind to protein A, to the mRNA of protein A, this is called the passenger strand. Okay, so this is called the passenger strand. And the one which can bind to the mRNA of protein A, that's called the guider strand, or just the guide strand. Okay, and what's going to happen is the enzyme Argonaut2 is going to break down the passenger strand. That's the first thing it's going to do, and then that will expose the guide strand. Okay, and we'll continue this discussion in the next video.